Welcome to the future of your genetic health and welcome to the Royal Society. Can I just check by a show of hands, how many of you have never been to the Royal Society before? That's pretty good. Yeah. The very quick counting Royal Society staff at the back of the room will have immediately noted it was exactly 63, if anyone's worrying. My name's Steve Cross, I'm your host for this event. Just to let you know how this event will go, we're going to spend a little bit of time chatting amongst our panel at the front, and then I'm hoping to open as much of this event as possible to you, to ask questions and make comments. When we get to that point, I'll run you through the rules of how we do that and still have a nice event together. We're due to run until 7:30, and we'll probably have to stop fairly quickly at 7:30. Can I just do another quick couple of demographic checks? How many of you in the room would describe yourselves as practicing scientists by a show of hands? Okay, we've got a few scattered around. Is anyone in the room a practicing medic? Good, a few of them as well, excellent. Do we have any students in the room? A few of those as well, brilliant. And is anyone in the room who was also at the day-long event that's been happening here about genetic technologies? There's a few of those. Okay, good, so we can tailor what we're going to say accordingly. So our panel are Robin and Sarah and Andrea, and what I'm going to ask them to do is to introduce themselves and their areas of expertise and a little bit about the topics that we're going to be talking about. So Robin, can I ask you to start? I will now sit down and make the whole thing less awkward.
Thank you. So I work at the Francis Crick Institute. I do basic science, so sort of medical research in aspects of genetics, stem cell biology and embryology. I'm also I'm very keen on discussing all these issues with the public, and in issues of how science generally is regulated and things like that. This particular topic, of course, I'm not a clinician. I don't work directly with human patients, but a lot of my work has helped. We've learnt from human genetics, and we've informed human genetics because we found several genes that have turned out to be quite important in respect to health in humans. So I'm interested in the topic generally, and I'm also interested because I've been given various roles. I'm chairing a Royal Society program on genetic technologies, which is actually looking more broadly at just human, but also plants and animals and the uses of the technologies there. And we're looking at all the aspects, the science of the way things should be regulated, how we engage the public, like we're doing this evening, and at the ethics as well. So.
Fantastic, thank you, Robin. Sarah.
I'm a researcher in bioethics at the University of Edinburgh. What that means, the bioethics part, not necessarily the Edinburgh part, is that I spend a lot of my time thinking about the sorts of technologies that Robin and his colleagues carry out in the lab, the sorts of genetic technologies that are affecting our health today or might affect our health in future, thinking about whether we should do them, what it means for society, how we're going to do them, how we're going to control them, those sorts of issues. So that's what my research is about. I'm also a member of the Royal Society's contact group on this theme.
Fantastic, thank you, Sarah. Andrea.
I'm a clinician scientist. I spend about 50% of my time seeing patients and the other 50% of the time running a very small lab. My particular interest is in neurological conditions and understanding how we use genetic information to try and treat neurological diseases, and I'm very fortunate to live in Oxford to do that.
Fantastic. I've had a little chat with the panel beforehand, and we've decided that we want this event to be about genetic technologies that affect humans as widely as possible. So I think we're going to be talking about everything from, I wrote a little list and I checked it with them because I haven't done any genetics for 10 years. We're going to talk about everything from genomic medicine to personally genetically tailored healthcare to directly altering the human genome. Is that all right? Good. And Robin, for people like me who aren't professional geneticists, can you give us a description of what is the state of the art? What is the science that is going to be underpinning our discussion today?
Okay. So several areas. So there is understanding what our genes, our genomes, the methods of sequencing DNA to read the code written in our DNA have developed incredibly rapidly over the last 10 years or so. So we have now these very high throughput methods, next generation sequencing, which can generate whole genome sequences in a very rapid period of time and very cheaply. And so we now have these huge projects, such as the 100,000 Genomes project, which is running in the UK, which will link the genome sequences, so all the DNA sequences of at least 100,000 people, with their health records and maybe lifestyle records, etc. So that's going to be a huge amount of information. We also have other methods to try and link or trying to discover the genetic causes of diseases or other traits. So there's a lot of work going on trying to understand, well, what are the factors that lead to correlate with, if you like, height or intelligence or other harder to define traits that we all have. Some of that gets quite contentious, but that's what's happening. So that's understanding what's in our DNA. So the methods there are developed very fast. We also traditionally we've had methods to change DNA in mammals for 30 years or so, but those methods have always been very inefficient, imprecise, therefore very unsafe. But in the last 6 years or so, we now have a set of methods, which are the genome editing methods, which are approaching the point where they are fast, accurate, and perhaps really safe to use in humans, both for trying to correct a genetic disease in a patient who's already suffering from a disease, and perhaps even to prevent our children getting a disease. So the latter is not being applied yet, but there is research going on about it. The former, so doing so-called somatic gene therapy, this is, if you like, altering genes within patients. There's already trials that have been launched and are being launched, and they're looking really quite promising. So that's the ability to specifically alter, in a very precise way, the genetic code to turn what was if you like, a bad version, a mutant version of a gene, back into a normal functioning version of that gene. So they're looking really promising, those methods. Then there's other areas which sort of feed into that, like synthetic biology, the idea that you can make organisms which didn't exist before at all, and for example, and then to introduce traits into organisms that you wouldn't have found naturally. And then I guess that leads into other issues, but we'll talk about those later.
We will, absolutely. Andrea, is there anything from the clinical side that you wanted to add to help us understand where we're at now in terms of technology and treatments and what's actually happening?
So I think one of the big areas is in gene therapy. There's been a lot in the news recently. You probably heard about Huntington's disease. It's not directly interfering with the genetic material per se, but the products of the genetic material, and that is incredibly promising. In terms of stem cell research, this is primarily still within the understanding diseases domain, so there's a lot less that's happening. There is some in certain areas, hematology and so on. So I think that's kind of where we are with the therapy bit. What we are really moving forward with very rapidly, as Robin has alluded to, is our understanding of how our genetic information tells the body how it does everything from how your heart beats to how your brain works. There are a lot of things we do not know and probably won't know for a long time, and that may well inform some of the discussion and some of the anxieties. For example, intelligence, which we think of as being this sort of unitary entity, is not in the slightest one thing. and understanding intelligence in a way that we might enhance it probably is going to take a very, very long time. What we are really, at the moment, understanding using the 100,000 genomes and other similar projects is specific genetic mistakes that cause very severe diseases in human beings, and how we can understand that to start to develop therapies. Now, a lot of the other stuff will probably come along in the future, but we're certainly not there yet. And things like the 100,000 Genomes Project have shown just how complex it is. And we're meant to be coming to the end of it very shortly, but it's been a lot more challenging than I think people thought a few years ago. So there's still a long way forward.
So given that all of these are gradual processes and things that have been changing for time, well, quite a bit of time, it's a question I have for all of you, but it's a question for Sarah first. And that is, the Royal Society are clearly putting quite a lot of money into this, doing public dialogues, doing events like this. There's lots of other organisations interested. Why are we talking about this now? And Sarah, that's for you, and then do just dive in as you feel like everybody else.
So, I mean, partly it is being driven by the science and recent developments, the clinical side of things, but I think there are also social and political changes implicated by these. So it's not just that we can do the science, but we are proposing to reorganise society in certain ways around our technological capabilities. Take genome sequencing and personalised medicine, for example. We recently saw the Chief Medical Officer's Generation Genome Report. The proposition that we're confronted with is that our entire NHS, our healthcare system, which is a moral and political, not just a clinical organisation, is going to be transformed by the promise of genomic medicine, that personalised medicine will become the way in which we organise healthcare as a social institution. And you know, that has huge potential, obviously, to improve health, but it also has certain implications about what we think of as the purpose of healthcare, the way that we make healthcare available, and so forth. In terms of the genome editing technologies, the fact that they are so easy to use, so widely accessible, the scaling up of that is meaning that they have different social effects. They are, if you like, they're not just confined to laboratories with specialised expertise, they're much more widely accessible. Added to that, I think, is a shift in, and this has been gradual, but I think it's a factor of all of these coming together, a shift in the way that we think about society and science and how we as individual citizens relate to science. So there's a lot of interest in ideas like the democratisation of science, being a citizen scientist, exploring what that means, exploring our role as active participants in science, whether that's as participants in research or people interested in science or people doing citizen science, wanting to explore what that means. And I think all of those factors have come together to mean that now is the right time to have this discussion.
Yeah. I mean, I can add that there's also there's a feeling that regulation should perhaps, that there's a need to adapt or change completely the way that a lot of things are regulated, because the regulation laws are usually very inflexible. The science is moving so rapidly that we think that, or many people are thinking that regulations need to be changed in a way that they can become flexible and cope with changes. So to regulate not the way you do things, but the specific aims of an application, for example, or basically just to accommodate these rapid changes. They have to be flexible. We also have the opportunities, perhaps now, to do things in the UK differently from the rest of Europe. I hate Brexit, but that's always gets raised as an issue. I'd much rather stay in Europe and change Europe than leave it. We also have to cope with the fact that whether we leave Europe or not, Europe is only one part of the world, does the rest of the world, and I think there's a feeling that because genetics is worldwide, things don't stay within countries. People move. There has to be some discussion at a very broad international level of how you can possibly regulate and control these things, because some countries, like the UK, have actually quite strong regulations in this area. Other countries have none. So that's an issue.
Andrea, why do you think we're talking about this now?
I think one of the key points, well, as we've already alluded to, the first is that genetic sequencing technologies have absolutely transformed our ability to understand individual bits of genetic information in a way that in my career we really couldn't do when I started 25, 30 years ago. And 10 years ago we really started to be able to. And the UK is without a doubt a world leader in this. So the 100,000 Genomes Project is the only one of its kind, and even in the United States and Canada, which have in some ways more funding to do this sort of thing, have not been able to organize such a big project with such a large population. So we have this incredible resource and incredible opportunity to understand human disease in a way that we couldn't before. And that has really piggybacked onto stem cell technology, for which there has been, again, a huge change in our understanding of how cells work and how we can manipulate them in laboratories, which means that we can make cellular models of human diseases in a way that we really couldn't before. So we were always dependent on rather unsatisfactory cellular models or gene-specific models that were very fiddly and expensive, or animal models which, leaving aside the ethics of using animals, don't really always represent human diseases as well as they might. And those were all problems that we were grappling with. So the gene editing or genome editing technology has really come just at the right time to make use of those other types of technology and come together in a way that we can look at specific gene mutations using whole genome sequencing, for example. We can then manipulate, using gene editing technology, those specific genes using stem cell technology, and then we can develop models within a laboratory that can tell you how those diseases work. And that has really opened the way for the first time to a much broader understanding of how we can use this for therapeutics. So drug development, which is, you know, when I started was the real driver 30 years ago. We could use genomes to develop new drugs. And yet it was really something that was rather out of reach for somebody like me. For the last 30 years, it's really been out of reach. And now we're at the point where we can genuinely say we can target specific genes and specific proteins and develop new drugs. And a lot of the discussion earlier today was exactly about that, and it was funded by AstraZeneca, presumably because of that. So we're really widening out what we can do in terms of therapeutics for the future.
Fantastic. And just to complete our understanding of the state of the art, we understand the science now, we understand a bit about why we're driven to talk about this. Robin and Sarah, I know you've both been involved with the survey to try and find out what people in the UK think about these technologies and where their thinking is at. Is there anything you can share with us from that that might help us understand the fact that technology and regulation and healthcare and need are all kind of hitting the same point together?
I mean, I could just start off saying this particular public engagement exercise, which has been published today, it had two strands to it. One was sort of deliberative focus groups where you have small groups of people, just under 30 people, that was in 3 different locations around the country, doing 3 different topics. But and those 30 or so people would get lots of information presented in an unbiased way. There would then be prolonged discussions amongst themselves and with experts in the room if they were there. They would then go away for 2 weeks, come back to another session where they would get more information and more debate, more discussions, having also in the intervening 2 weeks been given homework. They had to discuss these issues with their friends and families and come back with views from other people as well. And this is a very good way of doing having a public dialogue. It's costly, but it really gives you a nice sort of qualitative understanding of how people who don't have any scientific background at all, often how they look at these issues. There was also a survey, so it was an online survey of just over 2,000 people. That's always a little bit more limited because, of course, you can only provide a certain amount of background information for them before they answer questions. But having said that, we got fairly consistent results between the two processes, and there was a general high degree of optimism that genetic technology in general, so that's understanding DNA as well as manipulating it, changing it, can be of great benefit. Let's stick to the human health side, to human health and welfare. There were, of course, this optimism was nuanced or conditional around several issues. So appropriate regulation was felt to be very important to stop really bad things happening, but not to be too constrictive, that it would stop the science or stop applications of the science going forward. Because you won't learn unless you try something. That came from the public. They weren't told to come up with that idea at all. So generally it was really quite positive and optimistic. There were some other things, if you want me to carry on. So for example, they were asked about aspects of doing somatic gene therapy, so that's trying to treat a patient using genome editing, for example, techniques, versus making heritable changes, so manipulating the germline, so either very early embryo or cells that again give rise to sperm and eggs, to make changes that are potentially heritable. And the discussions around these areas in the focus groups was really very good. The survey also gave sort of actually very positive results. And the slightly surprising thing was that the public don't make any real distinction between these two. They didn't seem to care too much whether it was treating a patient or you were trying to use the methods to prevent a child being born with a disease. Now, I don't like giving public views like this to the public because you should be making up your own minds. But that was a very strong thing that came out of both methods that we used.
Sarah, did you have anything to add?
I guess I just wanted to share a little bit of my experience. I took part in the workshop sessions as an observer, and one thing that really struck me was how rich and nuanced the discussions out there are already. We have, as has been signalled, been having discussions about genetic modification for a very long time, and publics out there are grappling with these issues. Every single issue that I've thought about in my work as an ethicist came up in some way during the dialogue process. So people had not necessarily had the time to think about it in a structured way or to explore the arguments. But what was really valuable was having that time to do that with people in an extended process. In many ways, the survey results, it was 2,000 people, it's a barometer, it's a snapshot of a sort of general feeling. It's taking the temperature of roughly where we're up to. But to me, the real value of the exercise is actually in what's the genuine dialogue. If I had one wish about what could we do with this process, I would love for everybody in the UK, as a start, to have a chance to take part in a process like that. As I said, I'm a bioethicist. I spend a lot of my time thinking about these ideas. It would be great if everybody had that time to spend.
Yeah. Fantastic, thank you. Quick question for you, Andrea, as somebody working on the clinical edge of this. Everybody's mentioned so far that we've been having these discussions for a long time, and now they're slightly more sharp because of the things that are coming together, but they're issues that members of the public have been thinking about. Do you feel like these are new issues? Or are these the same ethical conversations you've been having for most of your professional career?
I think that they are, many of the issues are very familiar. So concerns about reproductive selection, concerns about what kind of traits are acceptable to adjust in some way. So this comes to the sort of concept of what is a disease, what is a disorder, what is a natural variation. Those sorts of issues. The whole issue about animals continues to be very pertinent. Mm-hmm. So all of those sorts of things are very familiar. And the other one that I was going to mention was drugs and clinical trials, so how we regulate these sorts of things being used. I think that we have been grappling with these in some way or another for a long time. What the genome editing revolution has done is really expand our horizons of what we can do. So that things that, as a scientist, 20, 30 years ago, we couldn't really imagine except as science fiction because they were just technically way too difficult, we can now start to imagine that we might do. Mm-hmm. Some of them are still incredibly difficult, and I think it's fair to say that CRISPR has really not come into clinical medicine yet. But there is the potential for it getting there, and that's, again, why it's an important discussion to be had now.
Sarah, you look like you have an ethical point to bring in.
Yeah, I think one of the game changers, especially with CRISPR, has been how it changes the risk profile around the use of these technologies. So we used to be able to say whether or not we were in principle in favour of or against human genetic modification. We all used to be able to say, but it's too risky. And, with genome editing technologies, we're going to get to the point pretty quickly, if we're not there already, that we might not just be able to hide behind this idea of it's too risky. When we started talking about this in the Hingston group 3 or so years ago now, we said there are these issues that will have to be addressed, off-target events, mosaicism. I'm sure Robin can explain more what those are if people need to know. But we said there are all of these areas of uncertainty that would need to be explored before this became ready for clinical application. In the 3 years since then, we have seen papers come out characterising off-target events, characterising the risk of mosaicism. We are very soon going to be at the point where we can no longer just say it's too physically risky, and then the question becomes, what's the moral risk that we're trying to mitigate? And just how bad is that moral risk? If we're going to take a technology and say, well, we understand the uncertainties, we understand what could go wrong, we know what the side effects might be of doing it. We know that the side effects, if you like, of not doing it are going to be that someone has a very, very serious disease or perhaps even a life-threatening disease. What is the moral risk that can be so bad that we're going to still say it's too risky in that circumstance?
Thank you. And Robin, you mentioned that the UK population are in favour of regulation as a way of dealing with this. I wanted to almost throw a spanner in the works for all of you. So if this technology is now at this incredible level, the UK population wants regulation, but we know that some places have no regulatory regime at all. There's a lot of changes happening in society. Can we regulate? Is regulation even possible? What challenges are there to the idea that if we put together the right regulatory framework, we can stop worrying about these moral risks? This is really one for all of you to dive in.
You can come up with a fairly simple list of sort of general principles of how this type of the applications could be, what you need to focus on in terms of how they should be regulated. I was involved in writing a report which was published by the National Academies of Sciences in the US a year ago. And this lists very clearly those principles. They're all fairly obvious, due diligence, care for individuals and all these things. And if you just have to, I think what we can do in the UK, because we're quite good at doing regulation generally, is to come up with a formula for how we feel we should regulate this, and then try and sell that formula to the rest of the world. We might get some objections, but we can at least try that. And then we are trying to involve other countries in the discussions. Again, part of the Royal Society is involved in, there's links with National Academies of Sciences in the US, the Chinese Academy of Sciences, China, and other organizations in other countries that are all interested in this topic. And so we're trying to extend the debate worldwide. Like Sarah, it would be great if everyone everywhere could be involved. It's going to be difficult, but just to try and bring in other countries, remembering that other countries often have very different cultural norms. So what we think may be a little scary here is not necessarily going to be scary in other countries. So that, yeah, Sarah, you want to say something?
We definitely need to be thinking beyond our national borders when it comes to regulation, but I'd sound a couple of critical notes, I suppose, or encourage people to think about it from a different perspective. I think we need to be careful of assuming that other countries don't have regulation or don't have good regulation. We know that the first 3 papers, I think, to use genome editing in human embryos were published in China, and I get a lot of people saying, oh, but they don't have any ethical standards there, who knows what they're doing? Well, actually, all of these studies went through ethical review. If you read the papers, and this is unusual for a scientific paper, if you read the papers, there is actual ethical commentary. The scientists are very well aware of these. So we do need to be a bit careful about assuming that in other countries there are no rules, and also in being a little exploitative sometimes about this idea that there are no rules in places overseas where we can go and do our work without, speaking as your sort of hypothetical exploitative scientist here. It's true that there are countries in which there is no regulation of genome editing, no regulation of human embryo research. Often these are countries where they're not doing any of that work because they don't have the scientific capacity. And I actually think where we get a real problem is where you might get scientists taking advantage of loopholes between regulatory systems to say, well, if I do this bit here and that bit there, and then say that there are no rules, then I can actually get away with it. And that's not that's the letter of the law, but not the spirit. We have seen an example of that happen with respect to mitochondrial replacement therapy. So most of you will know this is now legal in the UK after an extensive process of public consultation and regulatory reform and safety checking. So all of those elements that we want to see in place. Legal in the UK. The Institute of Medicine in the US has written a report saying, in principle, we think this could be ethically okay, although the FDA still doesn't allow it. The first baby born from this procedure was implanted into its mother's uterus in Mexico because the scientists said there are no rules, but that's actually not true. And he was an American scientist who went to Mexico to do the procedure. So I think that kind of scientific tourism is also something we need to be quite cautious about, and it has adverse effects that I will expound on if we want to get into that discussion.
Well, the other side of this, so we've got the regulation of the scientific research, but then you've got the regulation of accessing the kind of healthcare side of this, and what are you allowed to take part in, and who can take part in which bits of this revolution? So is that the other thing that we have to think about in all of this, the actual actors, the people looking for their own information or their own help? I mean, I'm looking at those of you who know people and know patients.
I think one of the concerns that certainly geneticists have is everybody going off in a sort of free-for-all and getting their genome sequenced, if you can afford it, with no information that goes along with it. So it's not so much, there's something about regulation here, but it's really more about information and informed choices. And actually, one of the things about the 100,000 Genomes Project that when I have been abroad and explained to people is that it's allowing equity of access and is very, very keen on encouraging people who otherwise wouldn't have had this access, not just to be able to have their genome sequenced for whatever reason, but to get a lot of information that goes along with it. So there's very clear genetic counseling that comes along with it. There's a lot of information on the internet that you can access, all the social media and so on. So clinical genetics as a speciality has really tried to take a lead in assisting people to understand what it is that they're getting out of their genetic material. So that's a really kind of important part about the information. I think that we are talking about pros and cons in a very, quite a controlled way here, because we are all somewhat making the assumption that everybody wants to be doing this for the good of humanity in some rather general way, and that people like me will do it for therapeutics and treatments, and then there's the problem of how there might be off-target effects, or some of the difficulties around who might access it, and equity and all that sort of thing. The other sort of elephant in the room really is that, leaving aside all of that, and I think something that lots of people are concerned about but don't necessarily raise, and I'll be interested to hear what you say, is that there are actually really bad guys out there who will be able to access this information in some way, in the same way that there are bad guys doing everything else. And however much regulation you have, actually you're assuming for regulation that most people are reasonably sensible, but are coming at it perhaps from slightly different angles, as opposed to the people out there who really might try and do something very bad with it. And we do need to have that in mind too, that we need to be clear about making sure that those sorts of problems are addressed, while at the same time very much making sure that we enable science to move forward in the beneficial ways that we know that this can do.
Sarah, I know you have strong thoughts on this point.
Yeah, so I mentioned earlier this turn towards really questioning the role of science with respect to publics, and questioning our role as citizens. And I think that one thing we've got to be very aware of is the turn, I guess, towards people wanting to have active agency with respect to science, to wanting to be active participants. One concern I have at the moment is that enthusiasm, that curiosity, that willingness to, and the desire to be part of science and to have part of science has to go somewhere, and we need to find ways to channel that through all of the great research that's going on at the moment. Because I think if we don't do that, and we've seen this happen in the stem cell field, people are going to find other ways to do experiments, to be scientific citizens in their own bodies and scientific pioneers in their own right. There are, as Andrea said, unscrupulous operators out there who will take advantage of this and say, come be in an N of 1 trial. We have no evidence because you are a unique individual. It's personalised medicine, and then they'll inject you with goodness knows what. You'll go away thinking you are part of science. It might or might not work. They'll never collect any information. That's not really science. So I think, in terms of how people are accessing these technologies, I think we need to make allowances for people's agency and to channel that in productive ways.
Yeah.
Thank you. I'm going to open this event up for you guys to ask questions now. There's a few things that I need to tell you. The first one is I'm in charge, which means if you want to say something, put your hand up and I'll point at you and I'll say, yes, you, please. The second thing is this is being videoed and live streamed and will go up on YouTube afterwards. So if you don't want your words to be on YouTube, don't say anything. That's the only way I can guarantee that you don't end up on YouTube. Maybe save your questions for a tweet or something later. And just on this thing of people accessing genetic analysis and things, I was wondering, has anyone in the room actually had their genome sequenced, has sent something off, or paid money and got things like that done? I did a project, good, somebody. I did a project 10 years ago where I took every single DNA test I could take without having to go through a doctor or genetic counsellor for a museum exhibition. And it's quite frightening, some of the stuff that people would send me with no genetic counselling. Also, I had to have a long conversation with my dad before I did it, because we had my Y chromosome typed. And the company said, there's a small chance you'll find out something you don't want to know, so you should talk. I don't think they know my dad, by the way. That's a general thing. So I'm going to throw this open to the room. We've got some fantastic Royal Society staff with microphones who'll be running around. Panellists, I'm not going to try and direct things to you individually. Dive in when you see anything you want to talk about. Feel free to disagree with each other the whole way along. So who would like to ask our panel a question or say something? I have a question right down the front. James, would you mind? Thank you for opening the questioning.
Oh gosh, my first time with the microphone. I have many questions, being a Star Trek fan, but I'm going to stay away from that.
None of these guys are in Star Trek, by the way. Just
I like it.
Will it be real? No. Just is there any hope of having an international regulatory body to, yes, I know scientists are better than politicians, and there's more hope that scientists will not act against the greater good. However, there's bad people everywhere. Is there any hope? Because I have a great deal of pessimism against how, the UN often tries to impose sanctions, and then the bad guy still does the bad thing. And I mean, I don't know who can really, maybe all of you can answer it, or maybe only one, but.
So is there any hope that we will have an international regulatory regime?
And how you see that possibly being structured at all?
Oh, so perfect. So if we had this, if the whole world together had agreed that we would have a unified regulatory regime, how would we have got there?
And how would you impose sanctions,
Yeah,
so to
how would we
speak,
enforce
for
it?
lack of a better term?
Thank you. Brilliant starting. Who would like to dive in on that one? The perfect future. How do we get there?
Sarah, yeah.
So you're right. We already don't have effective international regulation on a lot of things. I don't think there's a single thing on which we have complete international regulatory agreement. So I'm going to say we have to not be looking to those sorts of structures and mechanisms. I think we should be asking, why do we want something like this? What's the purpose of it? What's it trying to prevent? And we have to say, well, actually, you say, what would it look like? Well, would an international regulation of genome editing say you could or couldn't do it on humans? Would an international regulatory system for embryo research say you could or couldn't do embryo research? I think those are sort of questions that we're not going to be able to come to here are the rules and everyone must follow them. So I think we need to look more at process, at where in the process there's room for pluralism, for the different cultural views that exist both between sovereign nations and within sovereign nations. I don't think there's one set of rules that's going to work for everybody, nor do I think there's really a way to enforce that. So I think we have to be quite open-minded about what the content is, and instead think of ways that we can work together, be that as individuals, as different scientific communities, as different stakeholders, let alone different countries, to achieve what we want with genetic technologies.
Before that
international? I mean, I think it needs to be in international discussions. But you're quite right. I mean, there have been attempts, for example, to have a worldwide ban on human cloning. That never worked because it just never worked. It was never going to work. So that there's if you start off with acceptance, you're not going to get anyone signing up to a universal treaty to do this. And of course, it'd be very hard to get that to. Some countries will want more regulations, more specific regulations than others. You've got the whole issue of oversight, so it's no good just having regulations. You've got to have people checking what's actually happening. And then enforcing it, and that's really hard. I mean, it's hard. Countries like the USA, which is you think it's well regulated, actually in some areas it's very difficult. And the FDA, it's a very important organisation, but they don't have the resources to do the oversight. You're shaking your head. You know this. So it's a problem. But I think the more people involved in the discussions at all levels. We have, of course, in parallel with all this, we've got all social media and stuff. So if you can, empower people with social media to, hey, don't do silly things. Don't, you've got to be reasonable. Don't take unnecessary risks, but have proportionate regulation or ways of regulating things. Then there's some hope. I think I'm an optimist. I'm a terrible optimist.
Are we saying if we're going to get anywhere, it's going to be lots and lots and lots more talking? Beforehand, because
yes.
When you set me that question, my first thought was, if I put these three in a room, would they write a regulatory framework that they all agree on? And my first thought was probably not very quickly.
No.
So yeah, I think across the whole world might be a real challenge. Now, who else has a question for our panel? So I'm going to take the middle third row back. Thank you, Jamie. I've noted the other two.
Okay. Sarah mentioned that up to a point there would be physical risks to be addressed before a technology could have a certain impact, and then there are moral risks. moral risks and dilemmas. So I would know how to test for the physical risks, for example, off-target effects, mosaicism. You can do experiments to test them. How would you test a moral risks? How would you, in practice, devise maybe an experiment or a way for people to deal with this? You know, would it be like funding, you know, talking to the Black Mirror producers and do like episodes?
Do you mind if I ask you, are you a scientist? When you say I could test for the...
My background is as a scientist, so
Fantastic. Thank you.
Or
Sarah.
Yeah.
And
So
Robin, Andrea, do dive in if you want to.
Yeah. When I said there are moral risks, I think what I was trying to get at was that at some point us saying it's too risky is actually starting to be us signalling towards there are moral issues that we have not yet explored to the extent that we feel comfortable with, in the same way as characterising a physical risk requires us to explore those questions to the extent that we're comfortable with them. So, you know, we might not test them in even the same way. It's maybe a little bit of a false analogy to think of moral risk and physical risk in the same way. But you've brought up, I think, a really insightful point, which is that I think sometimes we try to guard against moral risks using physical or scientific mechanisms. We like to try and use science to answer everything, and actually to moral questions there often aren't scientific answers. Let me illustrate that with a couple of examples. One of the questions that came up in the debate over mitochondrial replacement was concern about identity, and people tried to answer that by saying, well, it's 0.01% of DNA, or it only contains 30-odd genes. But you know, when you're asking a question about the identity of your future child, you're not asking a scientific question, you're asking a moral and social question about kinship relations. So, you know, answering moral questions with science doesn't always get to the root of things, nor should we try to use science to guard against moral risks. And another example of this, we talked very briefly about genome synthesis. Some of you may have seen a couple of years ago now the announcement that a cabal of scientists, as they were portrayed, was proposing to synthesize a human genome, and there was some concern about this, particularly about the fact that there had been very little engagement or discussion before they came up with, hey, let's synthesize a human genome. And in response to this, the leader of the project said, well, we could engineer a sort of germline firewall so that any genome made in this way could never be used to create germline cells. But you know, engineering, however scientifically that would be done, engineering a germline firewall doesn't actually, to me, get to the real heart of the problem, which was there was very little engagement and discussion about this. You know, your germline firewall is not going to help that. So science, ethics, related, but you can't answer one with the other.
We do have some precedents. So in some of the technology, some of the potential applications of the technology. So I guess we. Even the more extreme one, which is the, if you like, more extreme, the one that people worry about the most, which is making the heritable change, the germline genome editing.
Or maybe don't worry about the most.
Or with some we don't worry. But you know, we've had pre-implantation genetic diagnosis, for example, which allows you to select an embryo that's not going to give rise to a child with disease. So we've had lots of discussions, and that's sort of understood and generally accepted. Genome editing, you can achieve the same thing effectively with that, and maybe more efficiently. You can use genome editing in situations where pre-implantation genetic diagnosis couldn't be used. So, for example, if one of the parents is homozygous for a dominant mutation, so it extends it beyond that, and then it extends it further, of course, because you can make changes which are, rather than selecting something that's already there, you have a particular combination of genes in different embryos. Of course, you can use the genome editing to make alterations to the genes, and that's where that's now stepping into an area that we've not been able to think about before, and that's, I think, one which really needs the proper debate.
Maybe to give another example, I mean, people are quite worried about who might start using this technology before we've had the full discussion. People are worried about enhancement, about cheating in sport. So let's say, you know, that some rogue scientist in a country somewhere else, because it will always be somewhere else and not here, rogue scientist in some country.
Rogue scientists in this country.
Well, yeah, rogue scientist. goes off, and germline engineers a super enhanced athlete who goes on and wins the Olympics. The child is fine, the child is happy, nobody is physically worse off. What's the physical risk of that? But we do feel there's some cause for concern around that. So that's an example of here is maybe what's the moral risk of this situation about which we're concerned. And to say that there may be a moral risk isn't to say that should never happen. It's to say we need to understand what those factors are that are causing us concern, hopefully before we go there.
Fantastic. I had another question just on the aisle. There's somebody wearing a beige coat. Would that be a fair description?
Sorry. So I'm not a scientist, but I would like to get involved from a citizen science perspective that you spoke about. I just wanted to know the different ways you can get involved or different things to check for. So I'm sure many people here are not scientists as well, but would like to be a part of this whole story. How can we contribute and how can we help?
Thank you very much. On my list was ask if there are things that people can do, so I can cross that one off. Brilliant. Thank you for reading my mind.
Well, you can order kits to do CRISPR if you want to.
You know, I said I know loads of rogue scientists. Don't become the rogue scientist.
No, but don't. Yeah, I mean there's sort of overlapping communities. So you've got the sort of do-it-yourself garage science community, and then you've got the biohackers. The biohackers want to actually modify themselves. The other community largely wants to, try and do some science in their garage. And that's generally, I think, pretty okay. There's a good community. They talk to each other. They sort of regulate each other to some extent. Some of the biohackers have been a little bit irresponsible recently in actually trying to, injecting themselves with CRISPR to, for example, try and increase muscle mass. They're now regretting it, by the way. Because it's silly. It's not going to work efficiently at all. In fact, they're more likely to do themselves damage. Again, it's having this debate at the right level and getting engaged by social media, if you like, other forms of engagement, just to spread the word that, please don't do something really stupid. You'll probably hurt yourself, but actually you also hurt the whole endeavor. We're trying to use these techniques to do some good things to help people who are suffering. Don't go and try and enhance anything, because it's going to spoil it for everyone if you mess things up.
Can I add to that? If it's not scientific, it's not citizen science. I think one of the big concerns about what's being marketed to people, and it often is marketed, is it's being sold as science, but what you're buying is not actually a chance to participate in science. You're just making the purveyors rich and not creating any knowledge. Here is something switching from DIY gene editing to thinking about genetic health technologies more generally and the idea of genomic medicine. Things that we can do are think about our role as patients in the NHS and citizens of this country and contributors to the sort of public health research that's going on. For many of you, if you go to your doctor or go to hospital to have a procedure, you may be asked if your data can be used for research, or if if you have a biopsy taken, there may be would you allow this to be used for research? Those sorts of things are hugely valuable. So, without population health records, we wouldn't have the understanding that we do now of how things like smoking and diet affect health. Without the use of biobanks made from stored tissue, we wouldn't understand cardiovascular disease the way that we do. The UK Biobank project at the moment, although that was recruited specifically as research, is currently transforming our understanding of all sorts of really complex health conditions. So one thing you can do if you get asked to participate in that simple way that just involves saying, yes, you can use my samples, or yes, you can include my data, say yes to that.
No, I agree with that. I think the biobanks are really, really important. They are expanding the way that they are being done. So current biobanks have not been using whole genome sequencing. They've been using more limited genetic analysis, and therefore the information you get out of it will be more limited. They are now starting to expand that out, and will be doing some whole genome sequencing. And it's relatively limited, but it will expand. Similarly, the 100,000 Genomes Project has a whole series of public engagement sessions on their website, so you can actually just go into the 100,000 genomes and then start to click on a series of links that can take you to all sorts of information. And one of the things about being a citizen scientist is being well informed, in the same way as being a professional scientist, you have to be well informed. And if you're going to be a member of the public, or you're going to be an amateur or a citizen scientist or a professional scientist, or if you're going to be a patient, you need to be very well informed about what's going on, whether it's your cancer or your kid's developmental delay, or whether it's understanding how genes work, so that you can have a sensible discussion about what these technologies can do. Then that's really important, because one of the things that, as you can hear, if you're injecting something that you think you're going to get bigger muscle mass by injecting CRISPR into your arms, the first thing is that tells you that somebody has absolutely no concept of how this technology actually works. And although we have spent the entire day talking about how amazing CRISPR is and how wonderful and efficient and everything else it is, the harsh reality is that we are doing it in the lab day by day, and it is actually incredibly difficult to get what you want to work in the way that you want it, in a way that will be medically or scientifically useful. It's not the sort of panacea, and getting away from that concept and using it in the way that it really does work is what's actually important.
Thank you so much. Now, I'm afraid I've run out of time for questions. Sorry, they only gave me an hour. I've increased your time by double than what's on the briefing sheet, but we've run out. What I would like to do is just ask each of the panel to maybe give us a minute or under a minute of your final thoughts. Now we've been all the way from the basic technology through creating a worldwide regulatory process to a whole load of people in their garages undermining the regulatory process by injecting themselves. What are your final thoughts on what we've been talking about that you'd like to share with us? And Robin, can I get you to go first, and we'll pan across this way, and then we will go back to our normal lives.
Well, I mean, I want everyone to think about the possibilities, and I don't think we should be too scared of things, but I think you've got to always balance risk and benefit, but think about things in, as was mentioned earlier, if you don't do something which could have been done, then that itself is an ethical, moral problem, because if you can stop people suffering, then probably we should try and do that. Think about where you think the limits should be. So we didn't really get into enhancements that much, but doing an enhancement is going to be technically very difficult. There may be some specific things you could do. I'd love to be able to see in infrared or ultraviolet. It'd be great fun, but that's we probably shouldn't go that way unless society wanted for some reason me to be able to do that. So the different types of enhancements. You've got personal enhancements where just that individual benefits, and then you've got enhancements that may be useful for society. And I gave an example this afternoon that if we want to go on long-distance space travel, we might have to enhance some people to be able to survive for long space journeys. If there's climate change causes a huge problem, we might have to start modifying ourselves. That is maybe a socially acceptable form of enhancement. I have no idea how you would enhance people for that, but just start thinking about where you would draw the boundaries and get involved in the discussions.
So Robin, long space journeys, and you want to see in ultraviolet and
yeah,
infrared.
you can tell. You see, I am a Star Trek fan.
Our Star Trek fan will tell us that's Geordie LaForge's visor in Next Generation, isn't it? Of course, yeah. No, we're not in favour of that. The Royal Society is not pro-Borg.
No.
Sarah?
In our chat before the panel, we were speculating wildly and perhaps a little irresponsibly about the future of genetic technology and what our health is going to look like in future. And I wanted to end by maybe proposing a couple of different ways that things might go and encouraging you to reflect on which ones of those we would or wouldn't want to see. With respect to gene editing, on the one hand, perhaps the utopic vision is that we have understood how to control it a lot better. We have controlled a lot of diseases that we currently can't cure. We have made sure that the technologies available in an equitable way. We have international harmony, even if not international uniformity on regulation, and things are generally going well. Maybe the dystopic vision is one where, look, well, we could have done all these things, but we didn't do them. We were too worried about regulating ourselves into the ground. Maybe we couldn't agree about it. Maybe we were too worried about the moral risks and did not even go there. And our descendants are saying, well, you could have found better treatments for cancer, you could have cured HIV, but you chose not to do it. Well, thanks for that. Great, guys, well done. So which of those futures do we want to see? With respect to personalised medicine and genetics, in one future, we are all, as part of the NHS, participants in a worldwide, perhaps, genomic research project. We understand as a result of that diseases better. We can get the treatments that work for us for our particular genomes. And again, attention to equity, attention to social justice, etc. In another, we've all gone, nope, it's my information, it belongs to me, it's mine, I'm not sharing it. I will sell it to certain companies, or perhaps if I buy a genetic test, they will both charge me for the privilege and then use it for research. But you know, public research, I'm having none of it. And as a result, all of the knowledge and all of the technologies are not there for us to benefit from. I know which one of those futures I'd rather see.
Thank you. Andrea?
These technologies are absolutely amazing. For those of us who are old enough to remember what science and medicine were like without them, there is no doubt that they are going to transform human health in a very significant way in the next 5 to 10 years and onwards from there. If you want to see that these technologies are being used in the best possible way, get involved as much as you can. Become as well-informed as you can. In clinical genetics, we spend an awful lot of our time informing people and explaining to them, this is what DNA looks like, this is what DNA does. You can do all of this online nowadays. You can get yourself well involved, support your genome project, support the Biobanks, support the NHS. These are all ways in which there is equity of access that in lots of places there isn't. And if you are well informed about the issues, then as the public, you will be able to provide a public voice for how those of us who are professionally involved can actually shape the future of science and medicine.
Fantastic, thank you. So we've now come to the end of our formal proceedings. There are a couple of things that I need to tell you. The first one is you've all got feedback forms on your chairs. It would be amazing and super helpful if you would mind filling them in and handing them in before you leave. The Royal Society genuinely use those forms to shape their programme. So if you've appreciated or not appreciated parts of tonight, you can help them choose how to run events in the future. The second thing is, if you have enjoyed tonight's event, and especially if you've never been here before, if you go to royalsociety.org, there are a whole load of other interesting public events that you can come and be part of. And the final thing I wanted to ask of you is whether you would join me in thanking our panellists for sharing with us for the last hour. Thank you very much.